So yeah, hello everyone. I'm happy to be with you today to share my work about how we can use deep learning to decode the tumor microenvironment. My name is Heba Sailem, and I am a research fellow at the University of Oxford at the Institute of Biomedical Engineering. So the reason I'm very interested in studying the tumor microenvironment is because it can have a significant role in patient outcome and response to treatment. The reason for that is that cancer cells interact with their surrounding, like they can interact with different types of immune cells like macrophages, which are known to have negative impact on cancer patients, while T cells have a more positive impact, but also with other cell types like fibroblasts and endothelial cells. And basically, this complex interaction can significantly determine patient outcome. However, because of these multiple play all can impact each other, having a way to decipher this interaction would be very important in order to improve therapeutic strategies or identify like reasons why the patient is not responding. And one attractive way to study the tumor microenvironment is using multiplexed imaging. So this technology basically allows us to image up to 60 markers in the same tumor simultaneously. And how this works, basically, we can use cyclic immunofluorescence, where we stain 3 markers and wash and restain again. And as you can see from this data set, we have 60 markers capturing like endothelial cells that can associate with angiogenesis, extracellular matrix, different immune cell types like CD68, CD8, as well as signaling activities like mTOR activities, phosphorylation of certain proteins, activity of key cancer pathways. So this gives us a lot of information at single cell level in the spatial environment. And to analyze this data, the first step is to segment each cell by finding the border of the nucleus or the cell. And from that, also, we need to determine the identity of cells, because we are dealing with intensity information that can be variable. We need to classify the different cell types like CD8 positive cells, CD68 positive cells, etc. From that, we can, for each cell, extract various information like the morphology, the length, the width, the area, but also the context. So how the surrounding of the cell, is it dense? Is it a lot of neighbors? What is the identity of surrounding cells, as well as the signaling, so the protein levels that we have in the data set or in the images. And when the particular data set I'm talking about today, we have around 3,000 cells per tumor, and for 746 colorectal tumors, this resulted in around 3 million cells. So this huge amount of single cell data that we need to use to understand patient phenotypes. So one way to study that is first to identify the phenotype of single cells. So using clustering, and here the single cells projected using t-SNE, we can identify what are the different signatures we have in the data set. And here I'm showing only the phenotype of cancer cells, so the ones that have epithelial origin. And from this, what we can then look at is the heterogeneity of the tumor, because each tumor will have different, many single cells that can have different phenotypes. So from here, we can cluster the patients based on how much they have from each single cell phenotype. So these patients seems to be less heterogeneous, while some patients will have many different phenotypes in the same tumor, which can have some relevance in their progression. However, these approaches can reveal interesting things, but does not account for the spatial information that we have from these images. So to address this challenge, I developed a convolutional neural network model, basically where we take single cell data from the images and construct a network or a graph where cells in this network, so the cells are representing the nodes, and the interactions or the edges between the nodes basically is based on spatial distance. So we set like cells that are close together probably have effect on each other, so we can connect them in the network. And what also we can do in this graph, embed attributes of like signaling, morphology, any other information we are interested to add to each cell. And how the graph neural network work is we have multiple layers, and we can pass the whole network, so the whole tumor information in this connected graph. And it starts by considering the state of the cell itself. So this is about, we start with H, and then update the state of each cell based on its neighbors. And how this happens is by considering the adjacency matrix between these cells. And also it's learn W through in order to predict certain outcomes. So to predict patient outcome, we will consider what are the influence of each cell on the other cells. And basically this gives us a very interesting machinery that allow us to consider the state of the cell, the state of neighbors in dynamic way to understand which ones are contributing to the outcome. And using this approach, I found we can predict patient outcome with area under the ROC curve of 83%, which is outperform existing methods. So another attractive aspect about using graph neural networks is that we can go back and look how different cells in the image contributed to the scores. So for each cell, we can assign it a score, whether it's associated with a positive class or high survival or low survival. And this can give us more information about the interactions or the cellular subpopulations that can contribute to ultimately to the outcome we are interested in. So to cut long story short, just from these analyses, what we found that basically looking at mTOR signaling in epithelial cells versus immune cells, we can see differential activity of mTOR. So mTOR is known to have negative impact on the patient, and usually it is targeted. So many treatments, many trials try to inhibit it because we know it have a bad effect on the cancer, however, or in the patient. So what my analysis showed based on this graph neural network results, that if we take the cells that are associated, like mTOR state in immune cells, it can be predictive of better outcome if it's negative in the epithelial compartment, because mTOR can be required for immune cell proliferation and differentiation. So looking further, I also found that in this particular class of patients that have better outcome, we see the reduced amount of macrophages, which is, again, the macrophages associated with worse outcomes. So this is consistent, as well as we see increased number of cytotoxic T cells basically in both groups that have immune positive mTOR. So this is very interesting because without this single cell analysis and determining the influence from the different cell types, these results might be missed. And of course, we can go further and look what are the signaling that is driving these differences between the groups. So we can see that the fourth group that associate with better outcome have like associate with EGFR pathway, as well as ERK, while the ones that associate with worse outcome and more mTOR activity in the epithelial compartment or cancer compartment associate with HER2 signaling, as well as various like invasion type like CD31 and CD68. So in summary, what I showed you that how we can use multiplexed imaging to study tumor heterogeneity and the microenvironment and the interplay between cells in the microenvironment. And graph neural networks provide a natural solution to the problem because we are considering the interaction, the spatial information, as well as single cell information. And what this study revealed that that mTOR can have a differential role in the tumor microenvironment, and this can have a significant impact when designing therapy because we can start stratifying patients based on their tumor signatures. So at the end, I would like to thank collaborators and contributors to this study, Fiona Genti, Claire Verrell, Mark Eastwood, and Muhammad Kawatmi, as well as my funder, the Wellcome Trust, for funding my fellowship, and CRUK. Thank you for listening, and I'm happy to take questions.
Yeah, we have a question.
Hi, Heba. I got a question to the individual cells in the image and then connect it to the prediction you made about survival of the patients. Were you also able to go back and identify the cells that were, because you said it's heterogeneous tissues, so there are also non-tumor cells in the original image. So were you able to make the prediction that, were you able to identify, for example, the macrophages or the neurons or the blood cells or something like that based on your glyph?
So basically, in this study, like we consider all the cells in the sample as tumors because it was a small biopsy. And yes, exactly, we try to go back and identify which cells are contributing and determining how their signaling is different. And the difference between doing that from single cell data straight away is that we might miss some complex interactions. So with the graph neural networks, we are able to better capture some the subtle interactions that is only possible by considering the spatial context of the cells.